Yes, and thank you everyone for getting up and coming after the great gala last night. It wasn't easy for everyone. So at Ocean, we're building genetic medicines for longevity. And just by way of background, we got started in about 2014, headquartered in Seattle, and our primary lab is in San Diego. And we're co-located with Entos Pharmaceuticals. And so that gives us access to GMP manufacturing. They're already a clinical company. So we've been able to accomplish more than we probably would on our own if we had to build all this out from scratch. But we have a really broad kind of vision and mandate for the platform. We think we can apply it to a lot of things in the field of aging and longevity. But we've kind of necessarily started developing treatments that you could get through regulatory agencies for a known indication and be attractive for licensing to pharmaceutical companies, but still act on one of these kind of primary drivers of the pathology of aging. So we had started in senolytics, which I think some of you here are probably familiar with, and our lead program now is in frailty, building muscle, and in the ablation of adipocytes. And given that this is a relatively short talk, I'm going to focus a little bit more on the science and the platform and how we're doing this, in hopes that it will inspire synergies and potential collaborations with all of you. And then I'll spend a few minutes talking about what we're actually doing with these lead programs as well. And so people have wanted to alter DNA ever since they knew what it was, but the Achilles heel of genetic medicine has really been delivery. And in the in vivo setting, at least, there's really two main players, these viral and non-viral vectors. And I won't go into all the details about them, but they all have known limitations, ranging from their payload, their toxicity, their immunogenicity. And we wanted to be able to go after much larger tissues that would require much higher doses and repeat doses. And so we tried to basically find a tool that would be the best of both worlds for delivery. So it would have the benefits of cost and scalability of the lipid-based vectors, but some of the entry characteristics of the virus. And this actually is made possible by a really interesting discovery in Canada. And I'm happy to nerd out about this later. You can ask me questions about it. It really is quite interesting, but I won't go too far into the weeds on it now. And basically, these fusogenic orthoreoviruses are the only non-enveloped virus known to science that has a fusion protein on it. And it is wildly different from an evolutionary point of view. It's about 100 times smaller than the nearest known fusogenic protein that's sufficient for membrane fusion. And you can see it there next to the HA protein from influenza. And the FAST protein down below, oh no, yeah, down here. Its ectodomain there is only about 12 amino acids long. And so this allows it to be effectively invisible to the immune system. And what it does when it gets close to a cell is that it will basically form a complex and it will mix the lipids of the particle with the lipids of the cell. Importantly, it doesn't need to be endocytosed. And that really allows it to get around this Faustian bargain of sorts that the LNPs in particular had to make with lipid chemistry, in that those all need to be endocytosed. They have to escape the endosome, so they're made possible by ionizable lipids. But those make them wildly toxic when they accumulate, and they're very liver tropic. So if you can take that away and you can rely now on the fusion protein for entry, it opens up a parameter space in lipid chemistry that's huge. I mean, you have thousands and thousands of lipids you can choose from. And normal LNP is about half ionizable lipid and 30% cholesterol. And so if you take those things away, you can do a lot of great extrahepatic targeting, but you also are free to alter the choice in lipids and formulation to confer a whole bunch of different kinds of tropism that don't require any interaction with receptors at all. And so you can load this thing up with basically any nucleic acids you want, or even combinations of DNA and RNA and complexes like that. But and it will deposit the cargo directly into the cytoplasm. Importantly, we also make this a single-step microfluidics process. So you can go full clinical scale. You can make it continuously, liters and liters of it. And the largest cost of goods for it is actually the DNA payload. And this is another benefit it has. It can deliver DNA as opposed to the LNPs that become very toxic with it. But it also takes advantage of something that the cost of DNA synthesis has been falling at a rate faster than Moore's law. And so it's a great confluence of technologies that I think would enable us to build a treatment that could address aging in a scalable and affordable manner. So when we started with this, the goal was to basically have a vector that had no tropism at all. We wanted to hit every cell in the body. And I mean, it's kind of a laughable target for those who focus on delivery. But that was the kind of design goals for it. And I said, since you're able to use these very different lipids, you can actually get something that's probably about as close as anyone's going to come for a long time. It's IV administration in a non-human primate. And you notice the first thing there is that the liver is no longer the primary uptake organ. And you can push this very far, in fact. This is an example of some of the different formulations that have been made both for some of our work and for Entos's. But the PLVs have an ability to stay where they're put, which is really useful. If you put something like an LNP here, I guess the takeaway is everywhere you put an LNP on the right there, they all end up in the liver. And whereas you can entirely detarget the liver with the PLV. And I always thought it was a little creepy that if you inject an LNP into your eye, it ends up in the liver in a few hours. But that's where we're at. And so this flexibility allows us to go after applications that I don't think would be possible with any other technology. And this kind of illustrates the reason why we were trying to do that back then. And this really got started when we were focused on senolytics. And you can see the mouse on the left has a tail vein administration of this reporter, CMV luciferase, and the whole animal lights up as you would expect from the prior data. But I think the really interesting one is this guy on the right. And that animal got the exact same dose and the exact same route of administration. But now the luciferase reporter is under control of a synthetic version of the p53 promoter. And we'd done this initially for an oncology project. And you can see there's a tumor implanted there in the red dashed line. And the expression of that luciferase is entirely restricted to the tumor now. So we didn't change the biodistribution in this at all. All those tissues got the same amount of DNA in both animals. But we're able to use basically information to target instead of chemistry. So combinations of promoters and repressors. And this, I think, is one of the most exciting developments in the field now as we get better and better at manipulating the control logic in life. But it also adds a great deal of safety. We have targeting based on the lipid formulation, the payload, and you can actually add surface receptors to target that way as well. We're not doing it for our programs right now, but our collaborators have made, probably 30 or 40 of them by now. Okay, so what we're going to do with it. Our lead program is building muscle. And the reason we want to do this really is that as you get older, strength is everything. And it not just affects your resilience against injury or disease, but your metabolic health, your quality of life, even your cognitive function. And so it's at this main axis that affects a lot of the problems with age. And so we want to take this into nursing home patient population that has a mobility disability. And an interesting thing has occurred over the last couple of years in particular, though, and that's the rise of all these GLP-1 agonists. And a reference there about Eli Lilly acquiring this anti-activin antibody, a similar pathway. And there's a real desire to limit or somehow attenuate the loss of lean muscle mass when you take these things, because it becomes very difficult to get back the older you are. So the genetic levers that regulate skeletal muscle growth have been studied for a long time, are pretty well understood. And that monstrous cow there was first characterized in the 1800s. And scientists have been busy manipulating these things for a long time. The mouse data there is relatively old. But you can see just how large you can get overexpressing follistatin. And the idea here is that myostatin is a genetic brake of sorts on muscle hypertrophy, and follistatin is you can think of it as the gas. When you exercise, the levels of follistatin go up about 7-fold for a few hours. And so we think that we can basically address this by turning it on all the time. And pharma has been working on drugging this for years, really. But I think the combination of technologies that were available didn't allow them to do what we can do today. And so we think we've been able to succeed with this. And so what we did was we took basically a nanoplasmid DNA construct that expresses follistatin. put it into the PLV, and injected it IV into these animals. And you can see here that after a single dose, in 6 months, they're 50% larger and twice as strong. And I don't think, for the record, it will be that extreme in humans. I think the largest anyone's done in a primate is around 30%. But what's great about this is both follistatin and the vector have already been used in clinic. So it's substantially de-risked, and I think actually will allow us to have a reasonable shot at going at this patient population. You can also use it locally, say for a cosmetic or for surgical type uses. And this is actually a new piece of data. The analysis for it's still ongoing, but we want to answer the question of could you administer this in really old animals and what would you see? And so these animals began the treatment at 2 years of age, followed them out until they're 2.5. And they didn't get enormous like the younger ones did, but they did have statistically significant improvement in strength. And I think that's what we're after. So the other program that we've been working on recently is for the removal of adipocytes. And in this, we're using a tool that was relatively similar to what we had built before for the senolytic. It is an inducible caspase-based suicide gene that's under control of an adipocyte promoter. And so we're going to take this into clinic initially for a rare disease called lipedema. And we have pharma who wants it for cosmetic use, so I'm happy to hand it off for that. I think it'd be quite lucrative, but I think it will also serve to validate that we can do this safely in a human. And so this is the disease we're after, lipedema. It's technically a connective tissue disease characterized by abnormal accumulation of fat, almost exclusively in women, usually in the lower extremities. And the only real treatments for it right now are repeat liposuction and life of ketosis. And so we think that we can effectively ablate these with a hypodermic needle. The initial proof of concept for this we did in tissue explants from patients who had tummy tucks. And we showed we could ablate, in that case, about 20% of the cells without causing any obvious damage histologically to the other tissue. And this kind of highlights that flexibility I talked about earlier with the PLV platform itself. You can see kind of where we started in terms of that first formulation down here was the one that I showed you in the monkeys, a clinical formulation that's broadly distributed. And we've been able to, just by changing the lipids of it, massively increase the delivery to adipocytes. And I think it just highlights really how much you can do when you're able to explore those kinds of variables. We just took this new one and put it back into mice, so injection, in that case, into the mammary fat pads, and showed that we're getting our reporter expressed both via in vivo imaging and later GFP. And so this is very much ongoing work. I want to save a little bit of time for questions, so I'll try to wrap this up. Like I said, we have a broad field of use for this, and we've started with things that we think we can get approved today, that we can get licensed today, but we have a very long roadmap and vision for the technology that goes into far more exotic things, and some things that the science isn't quite there yet to do, but we anticipated as we put this together that eventually the science would catch up with it and they would still need delivery. And so if any of you guys are working on these things, definitely chat with us. I shouldn't forget, we are raising money. We just opened our Series A. It has a lead from a large multinational pharmaceutical company. Actually, the paperwork just got approved for it a couple days ago. An amazing team. Everyone has lots of experience building biotechs and very well-established academic credentials. I think we're gonna go from there. I will say thank you to I know we have a bunch of actually our investors in the audience. I've listed some of the institutional ones here. Sens was actually one of the first investors in this, and Aubrey has got me into the space. So, and then credit to the people who are working on that aged mouse study as well. So, thank you very much.
Thanks so much, Matt. Any questions? We have plenty of time. There we go, in the middle. Who's got the microphone? There it is, coming.
After you remove the fat, if the individual does not change their habits, you're going to just end up with fat ankles or something? I mean, where does it go?
Well, the fat cells typically like to kind of grow and shrink as opposed to divide or die. And so when you kill them, you can get new ones, but they're going to have a hard time coming back. I think in the case of the local administration, you'd probably end up like someone who had liposuction, where you did not get the fat back where you got it, but you continued to gain weight elsewhere. Our goal with that longer term, though, is to go after visceral fat systemically, and which is kind of a different application. But yeah, the cells go into apoptosis and the fat's transferred out.
All right. Yep.
Yeah, thanks. Very interesting. Ultimately, could you be able to target individual cell types, for example, individual immune cells?
Oh yeah, absolutely. In fact, on the oncology side, we're working on in situ CAR T therapy. So we actually hit T cells inside the patient and cause them to transiently express a CAR.
Okay.
All right.
There's one more out.
Yeah, go ahead, please.
Cool talk, as always. Can you just talk me through a little bit more about how you said that you can target any tissue at all? How do you do that?
So there's like 3 dimensions of targeting. One is the formulation of the lipids, which give it kind of an affinity in general, a tropism of sorts for cells just based on the lipid chemistry. The promoter repressor payload is the other targeting mechanism where you can target them based on transcriptional activity. And then you can add surface ligands as well. So I didn't have time to show it, but you can attach targeting moieties to the C-terminus of that fusion protein, and it will cause it to be retained preferentially in the tissue.
All right. Thanks so much, Matt. Oh, wait.
Go.